Okay. Let's see. Let's talk some more about the example data that we were playing with last time, and then we'll see one more example data set today. Uh, Friday, we'll probably start in on the next topic of material, which I think the next thing we'll go to is next generation sequencing stuff. That's one particular type of technology that's widely used these days. And so we'll talk about some details that are specific to that type of data, and I'll show you some of the tools in R that are available in Bioconductor that are available to do statistical analysis with NGS, next-gen sequencing data. After that, maybe we'll do GWAS. I know there's a few of you who are interested in GWAS applications. It's another type of, another way to apply statistics to data like we have here. What are there any specific tasks in the context of GWAS that would be helpful to you? Like, I really wish I could learn more about predicting or FTRs or clustering, or in the context of what do you do on a day-to-day basis with your data?
I guess one of the biggest things is uh, calculating like proper support intervals for your peaks, the proper size of the GTO.
Mm-hmm.
So I know that you can't just use like the one bar interval because you need to adjust for size and population and all those other things
Uh-huh.
to adjust for it. But you could. So I'm interested in if you can apply basic tools to walls. I feel like you have to do a lot of steps before you run the analysis to do that.
Okay.
That might be outside the scope of this class.
Okay. Well, I'll incorporate that into picking what we talk about in the classroom. Yeah.
Maybe you have to do like posterior distributions beforehand.
Yeah. I'll try to keep our attention in the class as much as possible on specific topics that would have broad, interest to as many people as possible. Bayesian stuff, if we had to do a full Bayesian implementation, can be complicated in and of itself. We have a whole class on Bayesian statistics. So I'll look into it and think about it, and we'll definitely talk about GWAS for a while. And if you be thinking anybody in class who is interested in GWAS or any other specific technology, if there are specific things that you would like me to cover in the classroom, feel free to email me. When you do, give me as much detail as you can. I work with data like such and such. I'd like to learn how to do so and so, and then I'll try to balance it because we don't want to, take 4 lectures to address like one person's research project, but it doesn't benefit other people. So we'll try to get a good balance. But I would like to do whatever I can to make the content, as appropriate for you guys as I can. So just let me know. Missing data? I can make some comments on missing data. There are a couple of tools out there for dealing with them. You download one of these data sets and there's some NAs scattered around. What should you do? Should you delete the rows that have the NAs or delete the columns that have the NAs? Should you fill in the NAs with some number that you compute from the other numbers, or what? So yeah, I can comment on that. I wanted to go back to the example data we were playing with last time and kind of wrap up with them, and then look at one more example data set. Kind of the theme right now is let's learn how to explore data of this type, M by N matrix of numbers. And we've learned cluster analysis and principal components. We've also talked about doing hypothesis tests to go feature by feature, row by row, and ask which of my features are interesting, different between comparison groups. So these data that I'm showing you right now, and then the next ones that I'll show you, are just opportunities for us to apply those techniques and actually see what it looks like on real data. These data here are from mice, and remember we have obese or not obese, or lean mice, obese versus lean. And each mouse was either forced to fast overnight or not forced to fast overnight. So we have two factors for these data. We have obese, not obese, and feeding status. I've already read in the data. We log transformed the numbers. The first 5 columns or samples are obese mice. The last 5 are lean mice. And then the first 2 within each of those groups are the fasting mice. So I've also created a couple of factors, categorical variables in R speak, for obese lean and for fasting / ad lib eating. Okay. So we have 45,000 genes, and let's pick up where we left off. We did principal components. Let's remind ourselves what the output looked like. So there is a pattern that explains most of the variability in the data. And that pattern, we said, was to say create an index kind of variable that's a summary, basically an average of all the variables. Did I show you those simulations, those simulated data sets, PCA simulated data sets last time? Like I have some data where there's no structure. Here's what PCA looks like. Did I show you that? Okay, good. This is then indicative of all of these variables being correlated with each other. If they were not correlated with each other, and there was no correlation between the variables, there's no structure to the data. It's just all randomly bobbing around. And if there's no structure, then PCA will tell you that there's no structure. It will say the proportion of variance explained is just 1 divided by the number of variables you have. Because I'm going to divvy up, the best I can do is divvy up these explanations equally to all the P principal components. So seeing 90% says there's obviously a pattern that explains a lot. And because the variables are all kind of highly correlated with each other, positively correlated, they collectively are most informative. Average them up, basically. Okay, so I won't go further on PCA. That's the idea of PCA. It allows us to look at multivariate data and see what kind of patterns there are. Is there any kind of systematic pattern? And then interpret what those patterns are. Okay, now let's do testing. This is the part we did not do. I'm not going to emphasize this. Well, I'll show you what it is. It's not a big deal. We're going to go gene by gene, and I want a p-value. Let's test something. Let's filter out the genes in terms of are they interesting or not. And what we are going to consider, we have 2 factors. So I could ask a couple of things. I could ask, is there a difference for gene I? Is there a difference for gene I comparing obese to lean? I could also ask, is there a difference for gene I comparing fasting to ad lib eating? Does fasting have an effect on the expression level of this gene? Does obese lean have an effect on expression level of this gene? I could ask either one of those. We're going to ask the fasting question. So suppose the scientific interest is what happens at the genetic level, at the expression level, when you impose fasting diet on a mouse. Give me a p-value for the null hypothesis that there's no effect of fasting. So this that's inside this loop is a slightly more sophisticated example of doing a test for each of the features. We have already done a loop over all of our features where we have maybe just two comparison groups, treatment and control. Let's go to gene I and ask, is the treatment expression level different from the control expression level? If I wanted to do that, I could just do a two-sample t-test. T.test right here, store the p-value, and move on. We have two factors now, and we have then the opportunity to have a more complicated, or not complicated, but more flexible model for the data. So we could say, for example, that Yij is the expression, the intensity, the number in our matrix. So intensity for gene I sample J. I, in this case, goes from 1 to 45,000. J goes from 1 to 10. And we're gonna do basically a regression model for these data. I'm going to say Yij can be written systematically as an overall mean plus and kind of an effect, a shift in the mean associated with fasting versus lean. So let's create a variable called FL for fasting versus lean. Actually, let's make it F. Let's just make it capital Fi. And I'll define what that is in 1 second. Plus beta 2 times, this is like an effect of obese versus lean. So let's create another variable called OI. Okay, let's consider that as our model, plus random error. So this is a regression model. We say that response is equal to some linear combination of the variables. So it's an intercept plus beta 1, a slope kind of thing for variable 1, plus another slope kind of thing for variable 2. Plus random bobbing around, because if in the simple linear regression case, have in mind, if you were to have for each of these individuals a response variable y measured and some continuous x variable measured, draw a scatter plot, and you see maybe something like that. And regression is saying, on average, there's a line that relates x to y. But what we actually see for an individual is that line plus or minus these random deviations from the line. So those random deviations are those epsilon ij's. Standard way to write down a regression model. And everything just about in statistics is regression. ANOVA's regression, we can formulate most problems in terms of regression model. So here we have two factors, fasting versus not fasting. I have obese versus lean. Let's ask, is a gene different comparing fasting to not fasting? Okay, this model right here is a little different maybe from what you're used to thinking of in the context of regression, because this and this. Okay, what are these things? These are categorical variables. We'll say Fi is 1 if fasting. So if mouse I fasted. So it's a dummy variable. If mouse I fasted, then this variable turns on. Otherwise, it's off. So otherwise. And similarly, OI is an indicator for whether they are obese. Otherwise, then we can interpret these terms in the model then, like beta 1 is the mean difference for this gene, mean expression level for a fasting mouse minus the mean expression level for a non-fasting mouse among mice that have the same value for capital O. So adjusting for obesity, this is the estimated effect of fasting on the expression level, the mean expression level of this gene. This model right here then says, my parameter of interest would be beta 1. I want to do a test. I want to test whether beta 1 is equal to 0. Is beta 1 equal to 0 would be a null hypothesis, and if it were true, then this gene would be not interesting, in the context of which genes are relevant to this fasting response. If I don't get a small p-value for this gene, then I don't have evidence that it's interesting. This model, as it's written, is allowing or is restricting that the fasting effect is the same whether you are an obese mouse or a lean mouse. How could I make it so that we allow for the fasting effect to be different? How could I modify this model to make it so that there's a fasting effect for obese mice and there's a potentially different fasting effect for lean mice? Yes. So the regression idea that would be appropriate would be an interaction term. So I'm going to create a new coefficient, beta 3, and this multiplies the product of F and O. So that's called an interaction. And the purpose of putting that in there is so that now, like I was saying, there can be a separate fasting effect for the two obese statuses. That's the model that we're fitting in the R code. So how do we, quote, fit a model like that? LM in R is for fitting a linear model. It's how you do regression in R. So if you have a response and you want to model it in terms of a linear model, the right side of this expression tells R what your covariates are. So this that's highlighted says, I want you to have a term in the model for obese versus lean, a term in the model for fasting versus ad lib, and a term in the model for their product, which is an interaction term. So if we were to do this, pick a value of I, like I equals 21, and do this, we would have a fitted regression model for gene 21. I want to test whether there's a fasting effect, basically. And fasting, that F variable is in the model in two places because of the interaction. So just looking at, or just computing a p-value for this beta 1 would be insufficient. So I'm not going to go through the details of the interpretation. If you took 645 with me, then think about how would we interpret these model coefficients. Beta 1 is the mean change in expression for gene I comparing fasting to non-fasting. For who? In this model, I won't put you on the spot. If O is equal to 0 if you are lean, then beta 1 would be the mean difference comparing fasting to non-fasting for lean mice. Beta 1 plus beta 3 would be the corresponding mean difference for obese mice. We are allowing for there to be a different fasting effect for obese versus lean. If you want more detail on how to interpret one of these things, come talk to me. I'm happy to go through it. The point right now is, in order to test, is there a fasting effect? if I can formulate my problem in terms of a regression model, which I'm telling you most of the time can, then you can turn scientific questions like this one into a question about the parameters of the model. So in particular here, that question would correspond to a null hypothesis that I can pull F out of the model. If there's no fasting effect, then F doesn't need to be in the model at all. There is no fasting influence on this mean relationship. So another way to say that would be that the terms that correspond to capital F are both equal to 0. And we would compare that to the alternative that is not that. So that could be beta 1's not equal to 0, or beta 3 is not equal to 0, or they're both not equal to 0. There could be lots of different things. But the main thing I want to ask right now is there a fasting effect? And I need to test this kind of complex or compound null hypothesis. So I don't want to go too deep into this because it's not the point of the class, but that's what this code is doing. I'm going to go to gene I, and I'm going to fit the linear model that says the intensities for gene I, fit them in terms of a linear model with terms for obese lean, fasting ad lib, and an interaction. Now to test this null hypothesis, I don't just want the p-value for beta 1 for the reasons that I was saying a second ago. That's not sufficient. I need a more specific test for whether beta 1 and beta 3 are equal to 0. That's what I want to test. So it turns out you can do that with, there are techniques, statistical techniques for testing things like that. It's not a t-test. It could be an f-test. It could also be called a likelihood ratio test. And here's how you can do it in R. If you're doing regression, linear models, you can fit the model that you have in mind. I have in mind for this relationship, this model. There's an effect of fasting, an effect of obese lean, and there's an interaction. That's my model. If the null hypothesis is true, that would be a different model. So if the null hypothesis were true, the model I would think applies is the same one from above, except I pulled out fasting. I pulled out the F variable because under my null is that it's not necessary. So now I have two models that I have fit, an unrestricted and a restricted or null model. And then the ANOVA function in R will give you a p-value for whether those two models are statistically significantly different. Did you lose, a statistically significantly large amount of the fit whenever you removed fasting from the model? So here's the loop. It's going to go over all 45,000 genes, and it's going to pull out p-values. I guess the point of showing you this is that you're not always doing t-tests. That's not always the appropriate analysis method. If you're doing RNA-seq, for example, you're probably doing a regression model. If you are familiar with that stuff, you're doing a negative binomial regression model. So you're essentially looping over all of your features and getting a p-value, and you might get a p-value different ways depending on how the data were collected, what they represent. Okay, so this is just an illustration of a little bit more of a customized test that you want to do. Yes?
So when you would do this in your own research, you wouldn't take out both terms at the same time, right? You would take out the interaction first, test it, and then take out the next term. Dating them at the same time, you can't really say that which one is causing the significance.
So the question was about what we were showing in the script was to fit this model and then fit the model where both this term and that term have been removed. Okay, so there's a couple of issues that are relevant to that. One is I could ask different things. If I were to test, is the interaction significant? If I were to test whether the interaction is significant, then what that interaction term is the, let's just draw it. It will be worth the time, I think. So let's say here's fasting and, or let's say not fasting and not obese. Here is fasting and not obese. Here is not fasting but obese. And here is fasting obese. In terms of the model, what we're saying is that the mean, the mean for, suppose we did box plots like this. The mean for not fasting, not obese, would be beta 0, the intercept. Because we're defining fasting, F0 is for not fasting, and O equals 0 is not obese. So the way I isolate beta 0 and interpret what it means is to turn everything else off, which corresponds to not fasting, not obese. So that's what beta 0 is in this picture. The difference between a fasting mouse and a non-fasting mouse that is not obese. The difference between a fasting mouse and a non-fasting mouse that is not obese. If I focus on that specific obesity comparison, then this difference right here is beta 1. It's the fasting effect for lean mice. This is what we get by writing a model with an interaction term. So I won't go through everything, but you can go through this picture and figure out what each of these terms represent. So if I were to test beta 3, let's go ahead and do this, okay, because that will tell me what beta 3 is. Beta 1 is the fasting effect for lean. Beta 2 is the obese effect for not fasting. It is the obese effect for not fasting. So if I compare obese to not obese, not fasting, then this difference is beta 2. And then the fasting effect for obese patients, mice, fasting effect for obese would be this comparison, and this comparison would now be beta 1 plus beta 3. There's a fasting effect for lean, and there is a fasting effect for obese. So what is beta 3? Beta 3 is an interaction term. It is the difference in those effects. There's a fasting effect for obese, there's a fasting effect for not obese, and if you take that and subtract that, you're just left with beta 3. Beta 3 is the difference of effects. Is there an interaction? There is an interaction if fasting effect is one thing for obese and another thing for not obese. So if you went in and tested beta 3, is that equal to 0, you're not really testing is there a fasting effect. You're asking is there a difference in fasting effects? between obese and not obese. But if you're ultimately wanting to know, is there a fasting effect? You arguably are most interested in this as your null hypothesis. The idea of testing the interaction first and then the quote main effect it's been around for a while, and different people have different opinions on how to attack that. I'm a fan of just there's one question that this would answer the question, is there a fasting effect? I could go and ask more specific questions. Is there a difference? Is there an interaction? If there is not evidence of an interaction, let's say there is evidence of an interaction, beta 3 is statistically significantly not equal to 0. That means the fasting effect for obese is not equal to the fasting effect for lean. If I see that that's statistically significant, I really don't even need to go look at beta 1, because for there to be a difference of effects, there has to be an effect. So there is soundness to saying, let's start with the interaction term, test that. And if that's significant, you could go further, but you don't have to. If it's not significant, that doesn't necessarily mean there's not a fasting effect. It just means that there's not a difference in fasting effects. So then you could arguably say, now let's test beta 1 equal to 0. So yeah, you could kind of do a two-step process like that. And it depends on what you're interested in. Sometimes when you do an experiment you are scientifically interested in the interaction term. I think estrogen is good for your cancer safety, health. I also think diet is important, like the number of fruits and vegetables you eat, say. But I suspect that maybe there's an interaction between those two, and maybe there's something specific that we could learn about the interaction between the two. So I designed the study, and I want to do a test on the interaction term. I'm specifically interested in it. Sometimes you are, sometimes you aren't. Sometimes you're just including an interaction to allow for extra flexibility, because if you didn't include an interaction term but there was an interaction, then this beta 1 would be kind of an average across the levels that you didn't create an interaction for. I feel like I'm rambling. So we are going to get a p-value for each of the genes, and we are doing a slightly more customized way of doing a test. So that has run. Okay. I'm going to skip the volcano plot. It's really not that big of a deal. It's just a way of taking lots of p-values and visualizing them. But we know how to turn these p-values into false discovery rate estimates now, right? So let's make a histogram of the p-values. That's kind of a nice thing to look at initially, partly because it gives me a quick gauge of how much information is in the data, how much signal is in the data. Are there any differences that is there any evidence of this obese versus a fasting effect? Is there any evidence of a fasting effect? Yes or no? Do you feel like there's evidence of much of a fasting effect? How much signal? Do you feel like there's going to be a lot of genes that come out as highly statistically significant? Okay. I would say no. But the reason I would say no, you would say yes because there is a peak over here, right? I would say, relatively speaking, among bioinformatics data sets, if you make this picture for 30 data sets like this one will look like indicative of not much signal. There is a peak over at the left. Yes, that is true. And that's what we would be looking for. There are lots of p-values that tend to be small. You will almost always have some kind of peak at the left. But typically, you'll have a much bigger peak than this one. So this is saying that, yeah, there is a little bit, but there's not a lot. So that's one thing that I can look at in a picture like this. Another thing is, suppose this picture looks all crazy, which also happens not infrequently when you're doing data analysis with these types of bioinformatics data sets. You go get your p-values, you make a picture of the histogram or the histogram of the p-values, and you see that or something. Or you see that, or you see that. None of those are really feasible. If you're doing a test with a continuous test statistic like a t-statistic or a beta coefficient from a regression model, these are not what would be expected if things were going well. So it could be that there are issues in the data. If you make a picture like one of these and see unusual structure, for example, this kind of pattern can happen. You didn't get enough small p-values, as you would expect. Why would you not get enough small p-values if there's some different ways that can happen? One way is correlated data. If there's correlation in your data, it can really screw up the p-values. And if you've got bad p-values, you're going to have bad false discovery rates. So if you have a data set, omics type data, and you're going to do FDR and things like that, if you make a histogram of the p-values and you see one of these pictures, I would say don't try too hard to get fancy with your statistics, because it's going to be hard to convince somebody to believe them anyway. If your p-values look this very unusual, not according to a expected pattern, then something is amiss with the data. There might be correlated data, there might have been technical issues in the data, things like that. So I would be concerned if you have this type of thing. Okay, Q-values are p-value-like analogs that are specific to false discovery rates. You can get Q-values really easily using the p.adjust function. And then we might ask, are there any genes that had Q-values less than something small, like 0.05? And in this case, no, there are none. Here is a histogram of the Q-values. These are not expected to look uniform or anything. I don't have a specific expectation for these because they're not p-values. But you can see almost all of them are very big. They're all close to 1. So that says there's not a single gene that we can find for which we could say 5% false discovery rate. Okay, let's go to a different data set. So I think it's useful to see a few different examples because you can see what happens, what's typical and what you might expect to see with real data. So with our remaining 10 minutes, let's look at these data. This is Arabidopsis, small flowering plant. And here we have a two-class problem, a two-group problem where one group is treated one way and the other group is treated another way. So in this case, they were given auxin in one group, and in the other group, they were given auxin plus something else. For our purposes, it's M by N matrix of numbers. And we're going to go through the rows. We're going to look at structure. Look, can we see anything interesting in the data? So let's do clustering. Let's do PCA. And then we will do differential expression analysis to ask gene by gene, are you different comparing these two treatment groups? Now I could just do a t-test because it's just two groups. There's treatment basically and control. So let's read in the data. So we have 23,000 genes, 6 individuals. The first 3 individuals are, I think, auxin only, and the last 3 are auxin plus the other thing. Side-by-side box plots, always a good place to start. These look immaculate. Like we said, the mouse liver data looked immaculate, so I would bet that these have been processed somehow already. They've been normalized with some algorithm. Let's do cluster analysis. So how about Euclidean distance and complete linkage? I guess that's my go-to combo. H-clust and 2 pictures. Dendrogram on the left and then the scree plot on the right. Looking at the scree plot, I might say the last, or the place where things jump substantially is right here between this join point and that join point. So going from right to left, this rightmost point corresponds to the final merge event. So this rightmost point with a merge height of 140-ish corresponds to this joining that happened at the very end, 140-ish. So, okay, this difference between this point here and that point stands out visibly to me. It looks like there's a substantial jump at this point. So I'm going to keep this merge as well and call it necessary. The second merge was this one here. First one was from the top, or I'm sorry, the last merge, this final merge was here. Then there was this one. And I'm saying, based on that jump right there, that I believe both of those are reflecting real structure. So I'm going to kind of draw an imaginary horizontal line through this picture, and everything below that line, which includes this guy who technically dangles all the way down, everything that is disconnected below that line is now a cluster. So if I interpret the way I just said, how many clusters do we have? 3, right. So AB3 is its own thing, this is its own thing, and that's its own thing. What? So if, okay, so let's think about a couple of possible scree plots. If the scree plot looks like that, that's saying the final merge distance, so here's our dendrogram. Something like that. The final merge height is this height. And then to go down to where the next one was, we had to travel a long distance. That distance is, obviously big compared to how far you had to go to join to get the next one and the next one and the next one. So there's a substantial jump in terms of these distances, like this distance compared to that distance and so on. The fact that this is further up here compared to this being from there. Hard to explain. I look at the scree plot and I look for where's the obvious jump, if there is one. There's no guarantee that there will be one. I could see a picture that looks kind of just like that, in which case maybe I don't really believe there is any real clustering. But certainly if I have a picture like this one, that one dot that's obviously distinguished from everybody else says that the last joint was hard to do, kind of. Like the distance between the two pieces that you put together in that final one was substantially bigger than all the other ones that you've been piecing together along the way. So in a sense, this distinction, I believe is real. I don't maybe believe that this distinction, like those two from that one, those two from that one, I don't necessarily believe those are real. A difference between those three, I think those all belong together. Because the differences that had to happen to get them together were not very big. But in order to do this, it was a big distance that had to be traveled. So one dot that looks like the picture in the scree plot, I would interpret that as one real distinction, which means two clusters. So if there were two dots up here, now the big jump happened at this point. I would say that one distinguished real stuff, which means that's kind of where I'm going to draw my horizontal line that I was referring to. I could draw the line right there, in which case there's 1, 2 clusters. I could draw the line right there, in which case there's 1, 2, 3 clusters, and so on. So I'm trying to decide where to kind of draw this horizontal line. Hopefully that's somewhat helpful. You look at a scree plot, you look for, is there an obvious place where there's a jump? And then you figure out what that corresponds to in terms of where do I think there's real distinction and where do I just think everything's part of the same cluster.
So in that picture from the Arabidopsis file, would you be more inclined to think that that single one is maybe an outlier of that data than that it's in some grouping?
Yeah, I'd want to go look at it. I'd want to pull up the Excel spreadsheet with that data in it, and I'd do some detective work to try to figure out why is it different. Because it could be off here by itself because there's one outlying value in there. For some reason, a number that was supposed to be 2,000 got put in as 200,000. That would be enough possibly to make that whole column stand out as different, So I'd want to try to figure out what is different. Are all the numbers a little bit bigger than their corresponding numbers on the other arrays? Is this just more highly variable? Are there some outliers in there? Then take that to my collaborator and see if they know of any reason. Oh yeah, that was the one that we ran on Tuesday and the lab flooded that day or something, So if you can find a defensible story for why that mouse is that plant is different from the others, then it's fair to remove it, And you put in your paper in the methods section. We had 6 samples to begin with. 1 was removed because of such and such, and document it. What's not fair is to say, that one looks different, and if I remove it, I get a real nice looking picture. It's not fair to just pick to make the results look like what you want, obviously. PCA, let's do PCA real quick. I was also going to talk about the bootstrap, but I'm going to run out of time. So maybe we'll talk about it on Friday. The bootstrap is in your homework, but it's optional. So it's not written in the homework that it's optional, but I think did I mention that?
You mentioned it at the discussion that you might make it.
Okay. I did, okay. I intend for Bootstrap to be optional because I have not had time really to lecture fully on it.
Exam's still going to be Monday?
Exam is Monday.
How do you suggest what's the best prepping?
So best prepping for the exam, I will post an old exam. So I will do that today. I'll post an old exam, and Friday I will speak to what you can expect on the exam in some detail. But you do need a laptop with R because I'm going to ask you to download a data set and do something. These should all be good. They checked these laptops, so they are ready. They should be ready to go.
You don't mind if you use that person?
I don't mind. You can use your personal laptop. You can bring anything you want. You can access anything you want during the exam. So it'll be like, go download. Actually, this exam will not be all that similar to last year's exams because I have not given an exam through the computer before. I think it will work better because if I don't let you use your computer, it's kind of hard. I'm limited in what I can ask you, in terms of, questions on the exam. So this exam is going to be like these scripts. Go download such and such data. Here's a paragraph that describes the context. And maybe it could be like, carry out some exploratory analysis and tell me what you can find. So go make some box plots, do cluster analysis, do principal components, and interpret the results. I see in these data that there is apparently substantial, systematic differences between these two batches that were run. That's interesting. I would make a note and talk to my collaborator on that. Sample number 14 seems to be an outlier based on this picture. I looked at it, and the reason it's an outlier is because of this number right there or something like that. And I'll probably ask you, go do t-tests or something simple to get p-values. So given the context of the study, we had 3 comparison groups, and we want to ask, is each gene different across the comparison groups? Get a p-value for each gene. I might give you a tip on how to do it, like you can use this code to get a p-value. Convert that to false discovery rates, and maybe interpret the false discovery rate. You got 100 genes that had that corresponded to a false discovery rate less than 0.05. What can you tell me about those 100 genes? I can tell you I expect no more than 5% of them to be false discoveries. What if you had 200 that had p-values less than 0.05? Now what can you tell me about those 200? Might be like, let's separate the A plus from the A minus category. That's a harder question. But you might say, or you could say, I know, all I can tell you is that I expect no more than 5% of the null features to have been selected into that 200. But we talked about how that's of limited value because that can mean different things depending on how many null features there were to begin with. So I guess, download data, do some stuff that we have done. You can reference the scripts that are in here so you have some example code you could pull from. Interpret the results, like a clustering result, PCA, be able to interpret that type of stuff. Compute FDRs. And beyond that, it might be things that I maybe won't be surprised if everybody does not get them right, like the one I asked about FDR. I don't. So I would say focus on the scripts, running some of these examples and understanding what interpretations we are assigning to the script output and everything. And let me think about best ways to advise about the other content, but I'll stop rambling. So yeah, I don't intend for it to be too hard or take too much time, but as other people may know from last semester, sometimes I don't get the timing perfect. So if you run out of time or something like that, don't freak out. If it's hard for you, it's hard for everybody. I've said that a couple of times. 50 minutes, yeah. So I haven't written it yet, obviously, but I will work on it. Exam averages, it varies. It's so hard to write exams consistently hard, But mid-80s usually. So that's not bad, right? Okay.
We need to know all the underlying aspects of PCA, how what you should use the S matrix, B matrix.
I don't know, I might ask a question about PCA. Again, just my goal is not to give you a hard time with grades, but it is helpful to me to be able to separate. How much do you know? So some folks might be able to tell me, what's the variance of the first principal component? It's the eigenvalue of the covariance matrix or something like that. And I will reward them maybe with a couple extra points. I won't make that a huge part of the exam, but I reserve the right to ask a few kind of nuanced questions like that. So. About SVD? Yeah, I'm going to kind of officially de-emphasize SVD for our purposes. I could talk about it in the Q&A tonight if I have time. It's a lot like PCA. It's very similar. You won't lose much from the class if we don't do SVD, I think. I'm kind of leaning towards saying just do PCA. I think you can learn equivalent things. What's that? Yeah. Did I say anything about extra credit on the SVD part? I don't remember if I did. Yeah, okay. I'll talk to Yabu and I'll be fair somehow. We'll make it extra credit or something. All right. See you Friday.